This is not it. Can you see it?
Yes,
yeah, we
perfect.
see the PowerPoint.
Okay, perfect. Yep. All right. So, hi everyone. My name is Artemis. I'm a PhD student at the research group of our previous speaker, Tobias Lenz. And today I would like to talk to you about the variation in T-cell repertoire diversity within a teleost species. And I would like to start this with the short introduction, which I'm sure we don't really need, but nonetheless. So our story begins with when a pathogen encounters an antigen-presenting cell, and then it starts, well, as the name suggests, it presents pathogen-derived peptides on its surface. Then a naive T cell comes along and interacts with this complex. It undergoes clonal expansion, and depending on the nature of the T cell, it will either elicit cell-mediated immune responses or humoral immune responses. What I would like to focus today is this interaction right here, which is between the peptide MHC complex and the T cell receptor. And more specifically, I want to talk about the T cell receptor side of things, seeing how our previous talks were mostly focused on the MHC. Now we know that MHC diversity is very important in order to be able to recognize a vast array of pathogens. And once this happens and the epitope is presented on the cell surface of the antigen presenting cell, the second part of the story, which is equally as important, is the presence of a T cell with the right T cell receptor, which will be able to recognize this complex right here. And how does our body make sure that this T cell will be present? Well, for this, we need to take a look at the structure of the T cell receptor, which in this case, as we can see, and most T cell receptors are made of an alpha and a beta chain. We have a constant region and we have the variable region, which is the region which interacts with the peptide MHC complex. And how this region is made is in the thymus, and this is true for both mammals and other animals such as fish. During the maturation of lymphocytes, what happens is from a germline of TCR genes, multiple variable diversity joining and constant genes, we have somatic VDJ recombination, which means that a copy of each of those genes will come together in an almost stochastic fashion. And this rearranged DNA in the mature lymphocytes will start getting expressed and give rise to unique TCRs. So this is how a diverse and polymorphic TCR repertoire is generated. Now let's take a look at the numbers. It has been calculated that in the adult human body, we have more than 100 billion T cells, and most of these T cells express unique TCRs. We can visualize this like this here with a pool of different T cells, and a set of T cells with the same TCR is called a clonotype, as we can see here. And the entirety of different clonotypes constitutes the repertoire. Now, why is a diverse TCR repertoire so important? Well, as we saw previously, during antigen presentation, only when we have the right kind of T cell with the right kind of TCR will the adaptive immune response be activated. We can imagine a scenario where we have antigen presentation, however, we don't have the correct kind of T cell and therefore no adaptive immune response. And this we know could be detrimental to the host organism. And that is why it's important to look at the T cell receptor side of things. What we do in our lab is we utilize the 3-spine stickleback as a model system, and Tobias talked about it a little bit. It's a small teleost species living in the northern hemisphere. And lucky for us, it is readily bred and maintained in lab aquaria. And of course, it has been found to have an extensive copy number variation of both MHC1 and MHC2 genes. And this variation has been linked to parasite resistance, both in the wild and in experimental infections in the lab. Now, what is new is thanks to the work of our postdoc in our lab, Dr. Anna Telles, we now also have a TCR beta reference library of V, D, and J genes. As we can see here, this is the genome of the stickleback. In chromosome 15, we have these two different TCR beta loci. Each one has its own V, D, J, and C segments. Now, What we do in the lab is we use the Schistocephalus solidus system. This is a cystode that is trophically transmitted with a 3-host life cycle, as depicted here. I don't want to get into too much detail. What we need to remember for this presentation is that essentially an early life form of the cystode gets eaten up by these invertebrates called copepods, which act as, it's essentially food for the stickleback. So when a stickleback eats an infected copepod, it gets infected itself, and the cystode grows in its body cavity, as we can see here. And then the stickleback gets eaten by the bird, which is the definitive host, and the cycle starts again. So what is interesting about this system is that thanks to pioneering work from parasite physiologists, we can now culture it in vitro. So we can do experimental infections with it. And indeed, many such studies have been conducted in the past. And what we know is that this parasite grows very well, specifically inside the 3-spine stickleback body. It does not grow well in other fish species, meaning that it has to have developed some kind of avoidance of the immune response of the stickleback. Furthermore, we know that infected sticklebacks have enlarged spleens, which could indicate proliferation of leukocytes. We know that in the head kidney, we have lymphocyte proliferation in exposed but not infected fish. And we also know that S. solidus grows larger in sticklebacks with either low or high MHC diversity, but not intermediate diversity. And this points towards a certain role for the adaptive immune system in fighting this parasite. Now, what we have done in our lab is we have used lab-bred sticklebacks from 6 MHC2 typed families, with their MHC2 numbers varying from 2 all the way to 6 alleles. And we have 2 groups. We have a control group that is exposed to uninfected copepods, and we also have the test group, which is exposed to infected copepods. This experiment was done with valuable help from our technicians. And after 8 weeks of exposure period, what we have done is we have collected immunologically important organs of the stickleback, such as the head kidney, the gills, and the spleen. We have performed TCR beta targeted cDNA synthesis, library preparation, and sequencing. And also we have analyzed our data using a variety of different pipelines, which I can get into more detail later if you want me to. Now, let's take a look at what we have found after dissecting our fish at 8 weeks post-exposure. We see that for the 6 different families we have used, we have different infection rates, ranging from 15% all the way to 50%. And this is the MHC types and the sex ratio of the infected fish. We don't see any particular preference here. However, we do see that the infected individuals indeed have heavier spleens compared to control group. And what I would like to show you now, and I have to speed up, I realize, is that some preliminary results which we have gathered using split RNA from 4 individuals. 1 control individual from family A and 1 exposed but not infected individual, and again from family B, 1 control and 1 exposed and not infected individual. And this is the first set of results I want to show you. It's a heat map of the different V and J gene segments that have been used in the repertoires of the samples. And what we can see very easily for the V gene segment is that there's a strong bias towards 3 particular V segments as opposed to the rest. And also out of the available 13 gene segments, only 7 are used. So this does indicate a bias. When we look at the J gene segment usage, all 9 available segments are used. Again, there is some bias towards specific ones. What is interesting is that when we perform hierarchical clustering of using Euclidean distance of the samples, we see that the 2 control samples are clustered closer together compared to the other ones. And finally, when we take a look at the repertoire clonality, meaning how much of these clones that we find have undergone clonal expansion, if we take a look at the blue parts of the circles here, these are the singletons. It's our clonotypes that are occurring only once. And we can see that for both families, the control individuals seem to have a higher percentage of singletons. as opposed to the exposed individuals. And also, if we look at the second family right here, we can see that the high order clones, which are clonotypes that are occurring more than 2 times, 3 and up, take up a bigger part of the repertoire compared to the control group. So with this, I would like to get to the conclusions. We can see that the T-cell repertoire of these individuals reveals an adaptive immune system activation, especially when we compare this to some previous results we've gotten in the lab when we sequenced, young naive sticklebacks, and we found that up to 93% of the repertoire are singletons. We also can see some certain biases in the repertoire in terms of VJ's gene segment usage. However, we don't yet know if this is due to genetic reasons, such as the recombination, or due to some targeted response to the infection. Now, for our future work, we want to complete the sequencing of our library and then get into the in-depth analysis of the repertoires. We specifically want to answer questions such as how do exposed individuals compare to infected ones? Have they successfully managed to mount an adaptive immune response? Also, what is the extent of the overlap of different repertoires, and what is the effect of sex and MHC diversity on these repertoires? And with that, I would like to thank, of course, my group, my supervisor, all the technicians that helped in this experiment, and also you for listening to me. And now we'll take questions. Thank you.